Well, the thing is that I'm going to give an overview of some of the activities we have had in the Feed-a-Gene projects in Work Package 1. And I should say that the presentation here will only cover some of the aspects we have been dealing with in Work Package 1. And basically, it's just an overview of the activities in Work Package 1 in the Feed-a-Gene. Then I'm going to talk about alternative feed ingredients from European-grown soybeans, rapeseed meal, and green biomass. And then also talking about some of the technologies that has been applied. Basically, this is how the Work Package 1 that looks. We are all together 6 different tasks. The 3 first, task 1 to 3, they are very much on technologies. Then we have 2 tasks on nutritional evaluations, and a third one which are more on new methodologies for characterizing the nutritional value and composition of the feeds. When I modified this task, unfortunately, I also removed the partners in it, but on the task 1 up here is EFIB from France, Hamlet Protein that is involved. In task 1-2, it's Aarhus University and Hamlet Protein. In task 1-3, it's Bühler, EFIB, and Wageningen University and Research. In task 1-4, it's Aarhus University, Casper University, University of Newcastle, and EuroTier. And in task 1-5, it's EuroTier, University of Newcastle, DuPont. And then also the last one is Aarhus University and Wageningen University and Research. But basically what I'm dealing with is primarily some of these aspects we have up here, but I'm also going to address some of the data from the nutritional evaluations of it. If we're looking on the situation concerning the protein supply for the European market, you'll see that over the years, it's so that approximately 70% of the protein that's consumed within Europe, that comes from other countries outside the European Union. And basically, that's actually a proportion that haven't been changed that much over time. If you go all the way back to the late 70s or the middle 70s and onwards, you'll see there was an increase during the 90s, but then it has been dropping down again. So basically, it's a 30-70 split we have seen, and not really any improvements. And to some extent, it's due to political decisions and also agreements worldwide, but that is basically the situation as it is. If we also are looking on those which are the most important sources of protein in Europe, it's rapeseed, it's sunflower, and also soybeans, which actually have taken over after peas and beans, which in the past were more important than soybeans. But soybean has increased over the years and are now at a level of 2.5 million tons, primarily in the southern part of the main producer, that's Italy. You have the southern part of France, and you have into Romania in these areas. So that's the areas by which we see the production of soybeans, whereas rapeseed is somewhat more spread over Europe. So what we were looking at, that was also to look on some technologies to which we could process European-grown soybeans, and we have been looking on optimizing conditions for medium-sized crushers plant, which can be used for local and GMO-free soybean meal crops. And we are naturally interested to see whether we can create something that's also of nutrition and significant nutritional value, and also economical values in Europe. And what we have been looking for, that is a different way by which it can be processed, either by extruding pressing or pressing, cooking, or flaking, cooking, pressing, dehulling or not before the process. And there has been a number of different variations in terms of rate, speed, and so on in terms of the processing conditions. And basically, if we just are looking on the process in its grinding, we have a whole grain, which we then perform a dehulling, so that we have whole grains and also dehulled, which are then also processed by flaker cookings or by extrusion cookings. And that means that altogether we have produced in these processes that it's whole meal and dehulled meal with extrusion processing, and then we also have whole meal and flaker cooking process in whole meal and also dehulled. And basically some of the differences having the hull or not can have an impact on how efficient your thermal process will be in inactivating some of the anti-nutritional factors, and also on the extraction of the oils. And you can partly see that from the current slides here, where you will see that when you're looking on the proteins that in the products, that it's whether it's based on an extrusion pressing or dehulling or not, and also whether we have I should say that with regards to the extrusion pressing or the flaker cooking pressing, it has an impact on the oil that's present. I'm afraid this has moved a little bit. It should be the oil over here. Whereas when you come to the dehulling or not, it will have an impact on the protein content. But what also can be seen, that is when we have a dehulled product, we have a higher content of trypsin inhibitors left in it, which means that it could have a negative impact on the nutritional values. We performed in vitro digesting kinetics of the different products that was processed. And what you can see here, that is when we have the whole soy that haven't been processed, that it's much lower and are not coming to the same levels as with those which have been processed. But what we further can see, that is that the flaker cooking pressed products, it has a lower rate, and it also have a lower DH maximum for in vitro digestion, meaning that it has an inferior nutritional value relative to the others. And that is also what we saw when we were looking on the results we obtained in experiment with piglets, and where we can see that particularly this products that deviates to the negative side relative to the others, and also relative to a conventional soybean meal that actually was a very good quality soybean meal in having a quality as has been used by Hamlet Protein for their processing. And you'll notice that when you are comparing the digestibility, we are in some cases above what we have found for the conventional soybeans, whereas when you come to growth and so on, and some of these parameters, we are in the same order. Now to some of the technologies we have been working with rapeseeds. As I mentioned before, rapeseeds are the most important protein crops in Europe. But what we also know, that is that the digestibility of the nutrients in rapeseeds is lower than in soybean meal. And that is primarily, it's in terms of energy, but also in terms of protein and amino acids. And that is primarily because of a very lignified hull layer. And what further is so with the rapeseed meal, that is, or with the rapeseed, that is that a relatively high percentage of the protein and amino acids, they are associated to the hull layer. So they gave actually the idea to see whether it was possible with a form of tail end dehulling to fractionate the products that's from the crushers. And that is, well, some of the equipment that is used for these processes. And basically what we try to do, that is that we take the rapeseed meal after the extraction of the oil, and then separate it into a fine and also a coarse fraction. And from the data we have so far on the composition of the fine and also the coarse fraction from the process of rapeseed meal, we can see that we have obtained what we want to. We have a higher concentration of protein in the fine fraction than is the case with the rapeseeds as such, and also particularly also than in the coarse, and at the same time a lower content of fibers. So and actually the values for fibers here, they are close to what you will see also for soybean meal. These fractions are currently under evaluation in nutritional experiments. I haven't seen the data from it yet, but we naturally hope that we also can confirm that we have had a positive impact on the nutritional value of it. Moving on to the last part, which is more on, which are on the looking on the possibilities for taking out protein from green biomass. And I should say that basically it's started out as a local Danish interest in that field, primarily because we do have some environmental issues, as we have a lot of cash crops, primarily as cereals grown in Denmark. And we also know that we have a problem with nitrogen leakage of this to creeks, lakes, and inlets. And you could say the interest here is also to some extent related to the sustainable intensification, which is also an issue that is discussed in Europe. But basically you can also see that the whole Denmark, that is surrounded by water, so it is some of the problems. We have been looking on parts of it as a potential protein production, and if you look on some of the possibilities for using green biomass, it's some form of clovers, white and red clovers, and different types of grasses. And you'll notice that if we compare at least the potential production of protein within these plants, it's higher than we see with cereals, and also higher than we see with soybeans. But to be sure, there is also a number of problems that's not that easy to tackle. It is actually, we started early on in 12 and looking most on the plant production of it. And we have had national products that has been running, where we have been looking on it in a lab scale, in lab scales, and partly in pilot plant scales. And then we can say that for the Feed-a-Gene projects, it's so that we added actually to some of the local or national activities we had in our pilot plant facilities, enable us to do some more research in that area. But I should also mention that we have from decided in the end last year that we are going to build a demonstration plant which are going to to be in operation from 2019 and so on. So it's a gradual scale, but it can always be said it's maybe a little long shot. What we know from background on it, that is that when we are looking on it from our lab scale work, we can see that there could be an alternative protein source for monogastric farm animals, and our data we have obtained with when we do the evaluation on rat has actually been quite promising in that respect. And after several development, we for the last year of 2016, when we did the 17, when we did the extraction, we have a pilot plant that is more or less with this design. We are taking in the green biomass, we make a maceration, and we do a fractionation in screw press. We make a protein precipitation here by either heat exchange or fermentation, and then we make a separation, and we come out with a protein past here, which we then have to dry. What in addition, it's also so that the pulp in the Feed-a-Gene products, we have been testing them in experiment with rabbits, but in our country we are not producing that much of rabbits, and we are actually going to use it more either for cattle or for sows. And you can say what we intend to do, that is to remove the soluble part of the protein, which are not that much of use for the cattle, and use that for monogastric animals. That's the vision on it. However, we also have to admit that when we are looking on some of the data we have had from experiment with ileal cannulated pigs, they haven't been terribly promising so far. When you're looking on the standardized ileal digestibility of protein and amino acids from the green biomass we have, or the protein from the green biomass, you'll see it's somewhat lower to what we have in the soybean meal. And it's indeed lower than what we have found when we did the studies in rats. However, this year, and this is outside of what we have in the Feed-a-Gene products, we have managed by changing the way by which we harvest the green biomass, and also the way by which we perform the precipitation. Instead of using fermentation and also a heat exchanger, we have been using steam to precipitate out the protein. We have managed to reduce the ash content quite significantly, but what we also have found here, that is by reducing the ash content, we have managed to increase the protein content to something that is very similar to what you see in soy. However, these fractions haven't been nutritionally evaluated, but based on what I can see from all the other data, they actually seems to, or at least I would expect it to have a far better digestibility and compared to what we have been seeing in the previous studies. So a bit in summary, we have, we can say that with soy, with the process for soy, we have similar nutritional value as conventional processed soybean meal. Tail end processing of rapeseed meal, we have a fraction out of rapeseed meal with at least potential or higher protein quality. And in terms of the cracking of the green biomass, there is a potential new protein source for monogastric animals, but it will also clear that it will take some time before this will be on the market. Thank you for your attention.